Hello students. So first we study the C4 pathway or HSK pathway. So MD, H and C are slag. While working on sugarcane, found 4 carbon compound means dicarboxylic acid, and the first stable product of photosynthesis. It has been found to occurs in tropical and subtropical grasses and some dicotyledons. Some the important plants are sugarcane, maize and sorghum. So these plant which carbon dioxide fixation take place by Calvin cycle are known as C3 plants, because first stable compound, first product carbon dioxide fixation is made up from 3 carbon PGA. But in HSK pathway, first carbon dioxide fixation 4 carbon compound OAA, oxalic acetic acid. Hence such plants are known as C4 plant. So first, in certain tropical plants, carbon dioxide is not directly absorbed by rubisco due to lower concentration of carbon dioxide. They follows the another pathway in which first stable compound means here made up from 4 carbon molecules, 4 carbon atoms, that is oxalic acetic acid means OAA contain 4 carbon atom, hence is called C4 pathway. PEP is carbon dioxide acceptor, and the reaction pursued in this pathway are completed in 2 parts at 2 different sites, means 2 parts and 2 different locations. For example, first is in mesophyll cell and another in bundle sheath cell. So here C4 cycle or trans anatomy means C4 pathway in mesophyll cell. So this diagram number 1 shows the mesophyll cell, and C3 pathway in bundle sheath cell means here diagram number 2nd square shows the bundle sheath cell. C4 pathway completed in 2 steps. C4 pathway completed in 2 steps at 2 different sites. First step in mesophyll and another second step in bundle sheath of cell. Means here C4 path is completed in 2 steps at 2 different sites. First is in mesophyll and second in bundle sheath cell. So reaction in mesophyll cell, first fixation, carboxylation and reduction take place. But reaction in bundle sheath cells, second fixation means decarboxylation and second carbon dioxide fixation. So first in mesophyll cell, carbon dioxide taken from atmosphere is accepted by 3 carbon compound, and 3 carbon compound is PGA in chloroplast of mesophyll cell, leading to formation of 4 carbon compound, that is oxalic acetic acid. With the help of enzyme PEP, it is converted into another 4C compound, means made up from 4 carbon compound, is known as the malate acid. It is also known as the malic acid, malate meaning same. So it is transmitted to chloroplast of bundle sheath cell. Hence malic acid 4 carbon is converted into pyruvic acid, means pyruvic acid, here only 3 carbons are present. So converted into 3 carbon with the release 1 carbon. So here release 1 carbon because malic acid is made from the 4 carbon, and if inserted in bundle sheath cell, so that time they converted into 3 PGA pyruvic acid. Okay, 3 carbon atoms are here and 1 carbon. Okay, so remaining 1 in cytoplasm. Okay, so here malic acid 4 carbon is converted into pyruvic acid 3 carbon with release of carbon dioxide in cytoplasm. Thus, concentration of carbon dioxide is increases in bundle sheath cell. Concentration of carbon dioxide is increases in bundle sheath cell. So, chloroplast of this cell contain enzyme of Calvin cycle. Because of high concentration of carbon dioxide, RUBP carboxylase participate in Calvin cycle and not photorespiration. So sugar formed in Calvin cycle is transported into the phloem, and pyruvic acid generate in bundle sheath cell re-enters mesophile cell and regenerate the PGA by consuming ATP. Since the conversion results in formation of AMP, not ADP, two adenosine triphosphates are regenerate to regenerate ATP from AMP. So this C4 pathway needs 12 additional ATP and C3 pathway needs 18 ATP for the synthesis of one glucose molecule. Important point is here. C4 pathway needs 12 additional ATP and C3 pathway requires 18 ATP for synthesis of one glucose molecule, where C4 pathway requires 30 ATP. Thus, C4 plants are better photosynthesis and there is no photorespiration in these plants. It's about the crans anatomy. So meaning of the crans meaning the width or necklace. So leaves of C4 plants show some structural peculiarities are called crans anatomy. Means mesophile is not differentiated into plastid and spongy tissue. So first, mesophile cells. So first point in a mesophile cell, undifferentiated means only one type of cell. Compactly arranged cells. All mesophile cells are compactly arranged. Chloroplast is granule in mesophile cell, smaller in size, more in number and very less stomata present in mesophile cell. And enzyme is PEP carboxylase present in mesophile cell. So first, C4 plant shows the crans anatomy. Mesophile tissue is not differentiated into plastid and spongy tissue. Mesophile cell with granule chloroplast and bundle of sheath cell with a granule chloroplast. Vascular bundle is surrounded by bundle sheath cells. And enzyme is PEP carboxylase present in mesophile chloroplast, but remaining enzyme, that is RUBP carboxylase, is present in bundle sheath cell. So remaining portion is known as the bundle sheath cell, And bundle sheath cells, vascular bundles are surrounded by bundle sheath cells. And vascular bundles is made up from xylem and phloem. So bundle sheath cells are radially arranged, and chloroplast is a granule, means chloroplast without grana, bigger in size and less in number. An enzyme is Rubisco, means RUBISCO, is bundle sheath chloroplast. Okay, so here PEP carboxylase present in a mesophile, but RUBP carboxylase enzyme present in bundle sheath cell. RUBP carboxylase is also known as Rubisco. So Rubisco enzyme present in bundle sheath cell. Chloroplast in bundle sheath cells are dimorphic. So meaning of dimorphic, di means two. So here two types of chloroplast present in Kranz anatomy. For example, in mesophile cell and in bundle sheath cell. In mesophile cell, a granule, in mesophile cell, granule small with grana and arranged centrifugally. But in case of bundle sheath cell, granule, a granule large without grana arranged centripetally. So this is the only introduction about the mesophile cell and bundle sheath cell with chloroplast and enzymes. So here, after the Kranz anatomy, next point. So here, diagram of Kranz anatomy. So here, diagram is very important in our examination, because explain the Kranz anatomy, this point gives to the 3 marks and explanation gives to 1.5 mark, and remaining 1.5 mark for correct labeled diagram. Means your diagram gives to 1.5 mark. So half mark for proportionate diagram and remaining 1 mark for any correct two labels, and remaining explanation gives to the 1.5 mark for this question. So here diagram, tiers of leaf showing Kranz anatomy. So upper side located layer is known as upper epidermis. Below upper epidermis, small cells are compactly arranged, is known as mesophile cells with granule chloroplast. So green colored compactly arranged cells are known as the mesophile cells, and in mesophile cells granule chloroplast is present. So here center two flower like structures, and at the center made up from xylem and phloem, means vascular bundles. So vascular bundles means xylem and phloem. So surrounded by vascular bundle, a flower like structure is known as the bundle sheath cell, and bundle sheath cells with a granule chloroplast. So a granule chloroplast present in bundle sheath cells, and lower side layer is located, is known as lower epidermis, and on a lower side one opening is present. Here in diagram shows only one opening, and that opening is known as the stomata. Okay, so here we discuss about the tiers of leaf showing Kranz anatomy. So important point is upper epidermis, mesophile cells are related with the chloroplast, then vascular bundles means xylem and phloem surrounded by bundle sheath cell with a granule chloroplast, and last layer lowermost is known as the lower epidermis with stomatal opening.